Paleoproteomics is a study of ancient protein preserved in archaeological, paleontological, and historical samples. Proteins are large molecules made up of chains of amino acids linked by peptide bonds. Amino acids are the basic building blocks of proteins that contain both amino and carboxylic acid functional groups. Peptides are a shorter chain of amino acids, and in the context of ancient samples, intact full-length proteins rarely survive. We typically recover only fragmented peptide for analysis. In our laboratory, we use the sp3 extraction protocol, which employs magnetic beads to capture and purify peptide from complex mixtures. This method efficiently removes contaminants and is especially effective for the low abundance degraded peptide typical of ancient samples. Proteins are enzymatically digested into peptide, which then bind to the bead, allowing for cleanup and concentration before analysis. The extract peptides are sequenced using LC tandem MS, producing spectra that bioinformatic tools analyze by matching peptide fragments to protein databases. Depending on the analyte, different software like MaxQuant, Peak, pFind, Sage, or NovaCloud can be used to analyze proteomics datasets. This tool facilitates the identification of ancient protein sequences, detection of post-translational modification, and extraction of taxonomic information, offering valuable insight into past organisms and their environment. Proteins complement DNA by providing information that goes beyond the genetic code itself. While the genome represents the complete set of genetic information encoded in DNA, the proteome is the entire set of proteins expressed by an organism, tissue, or cell at a given time. Due to the degeneracy of the genetic code, the proteome theoretically contains less genetic information than the genome. However, ancient proteins offer insight into gene expression and functional biology that ancient DNA cannot directly provide. For example, the protein reflects the active translation of genes, revealing aspects of physiology, phenotype, and environmental adaptation. They also allow identification of organisms with RNA genomes, such as ancient viruses. Finally, ancient proteins also allow the study of host-immune responses to past infection by detecting immune-related proteins or antibodies, providing insight into how the immune system evolved in response to ancient pathogens. Moreover, studying ancient proteins is not only a valuable complement to ancient DNA analysis, but also a crucial tool when DNA is no longer preserved. Ancient DNA is highly sensitive to environmental degradation and, except under exceptional conditions, rarely survive beyond a few 100,000 years. In contrast, proteins are chemically more stable and often better preserved in archaeological contexts, particularly in mineralized tissue, such as bone, teeth, dental enamel, calculus, or eggshell. Due to their greater stability, proteins can be recovered from material dating back to the Pleistocene and even as far as 20 million years ago. Together, these advances highlight paleoproteomics as a powerful and expanding field that extends our understanding of prehistoric eras, shedding light on extinct species, but also on host immune response, ancient dietary practices, and even RNA-based organisms.